today we're gonna address how to use it. I will start this day with telling you a little bit on how to prepare your surface of microfluidics or nanofluidics to accommodate your applications, whether that would be biocompatibility, whether that could be, you know, designing surface charge, for example, that you're looking for, doing, for example, microcapillary electrophoresis, or preparing surface for processing biological samples and maybe how to prevent non-specific adsorption. So this clock here is really off, 9:09. Majority of research in the center, in our lab and in the center, rotates around those four thermoplastic materials. Those would be polymethyl methacrylate, which is just acrylic. You can see a lot of furniture made of acrylic. That's the same type of material, PMMA in short. Polycarbonate, cyclic olefin copolymers, and you know that there could be different grades, so to speak, of the cyclic olefin copolymer. So when you're reporting or when you're looking for your application, pay attention into those different type of copolymers, and you can see that they vary concentration of the norbornene content. And there are very interesting consequences as a result of this different composition of the norbornene and polyethylene. And there is a family of cyclic olefin polymers, COP. And again, these go, the COC, it's mainly used, trade name is Topaz, when you hear. And for COP, that would be Xeonex. And again, you have a different grades, different, what we're going to mention a little bit about it, different glass transition temperatures that will affect how you use those thermoplastics in your application. So I'm not going to go through all of it. You have all the material. In my presentation, you have also a strong sort of appendix section with additional information that I encourage you to take a look at. So thermoplastics, by definition, those are the materials that are softened or melted by the application of heat and can be processed. We've heard about this yesterday from Dr. Park's presentation. We can process them and fabricate devices from those thermoplastic by thermoforming or extrusion or injection molding. And you also have heard that. So depending how much heat actually you're going to apply, then you can use those two different methods. When I do the hot embossing, I do not melt the polymer. I just go above the glass transition temperature where those polymer chains start really soften, and then you do the imprinting. When you do injection molding, you do go to the melting temperature, so you have the thermoplastic in a liquid form. The bottom line and relevant for what I'm going to say today is that the thermoplastics that belong to this amorphous polymer category, as you can see here, and they're listed here in this pyramid, you use those thermoplastic below their glass transition temperature. They have to be in a rigid form. So when you're designing certain thermal processes on your microfluidics, for example, a long time ago we started working with amplification PCR on the microfluidic chip. You need to be wary of your 95-degree step in the amplification and match the thermoplastic material. So we opted to go with polycarbonate that has glass transition temperature of 150 degrees C. So it's well above that 95-degree thermal step that's required in the processing. So that's how you match. Dr. Soper said, we have all those variety of the thermoplastic, and you can match the thermoplastic to your application. That's how the level, the glass transition temperature is one of the parameters that you need to consider when you're doing your when you're choosing your material of choice. So a lot of modification, surface modification, for example, some kind of covalent attachment of an agent, we do perform when the microfluidic or nanofluidic is actually enclosed. So imagine when you fabricate let's for the sake of this conversation to microfluidic. When you fabricate, I will tell people, you fabricate a channel, right? You have a channel, microchannel. You need to form a tunnel to actually perform the processing, right? You need to put a roof on top of that channel to create basically enclosed structure. So before we, in most cases, we perform the modification after we bond and assemble the device. And one, the most often used method in our lab is what we call thermal fusion bonding, where you have, we call it monolithic assembly, means your cover plate and your substrate material are made of the same material, thermoplastic. They have the same glass transition temperature. When you bring them in a close contact, apply pressure and apply the temperature slightly above or at glass transition temperature. Remember, those polymers from the substrate and from the cover plate, they start wiggling. They become interwoven, and then you cool it. And this gives you a very strong bonding. It can withstand up to 100 PSI pressure that is absolutely fitted for numerous applications. So that's how you do it. You can use simple convection oven if you don't have a thermal press. We've done it for a long time, basically, and I'll be happy to show you in the lab. You place it between two glass plates. You use bonding clips, and you assemble your device. It was a bit of an art at some point. Therefore, we moved to the more precise way of doing this using press, and you'll see it today. But there's nothing wrong when you want to start your research with this kind of in-house type of assembly. So when you think about the modification of the surface, and when you think about the surface, you need to consider how that interface gonna be looking like between your thermoplastic and the liquid and the buffer and the sample. And the measure of it is the surface energy of the plastic, where that basically will dictate how this interface those interactions at this interface are taking place. We know that the lower the energy, it's much harder to wet the surface, right? It's hydrophobic. There are applications where you may work with hydrophobic surface. You can design those surfaces to be really hydrophobic, right? To prevent some non-specific adsorption of the aqueous-based solutions. But in majority of our applications, we actually want the surface to be hydrophilic. We want a lot of interactions of the biological fluid or the buffer or whatever standard solution to actually actually interact with the surface. So you can see that here you have a surface energy. Those are low surface energies, means this is hydrophobic surface. How do I measure this in the lab in an easy way? There's a very simple way to do this, is by measuring the water contact angle. And by evaluating the shape of that liquid water, of that water drop, you can tell whether the surface is indeed hydrophilic or hydrophobic. Think of it as a, elephant plant or any plants when they get wet and the water drops, right? You have those perfect drops. Think of a Teflon, super hydrophobic. You'll have a perfectly round drop if you deposit that water on the surface. That means it's a very high, based on the tangent, how you draw the tangent of this water contact angle, you're going to end up with very high water contact angles. Low energy, high water contact angle means hydrophobic surface. High energy, low water contact angle tells you it's a nice and hydrophilic surface. And we'll show you that also in the lab, so you have a first-hand experience on evaluation. So how can I make those thermoplastics hydrophilic? And there are a few options. One is using oxygen plasma, and it's a very dynamic process. There are a lot of positive ions, electrons, a lot of active species in the oxygen plasma. Don't be mistaken, it's not easy to work with actually oxygen plasma. You need to make sure your flow of oxygen is constant, and you know exactly what that is. You need to make sure that the energy of this in the setup for the oxygen plasma system is constant. The position from the plasma of your substrate. So this works, but you have to be really careful. It can be sometimes irreproducible. I could, for example, it's a normal way to oxidize PDMS and glass and bring it, for those of you who work with PDMS, and it bonds instantaneously. And sometimes you have your flow conditions of oxygen too high, and you will never bond this. So be aware that it's not a simple procedure to use oxygen plasma. And when you do, you need to report exactly conditions. What we really like in our lab to use, which we believe gives us much more reproducible results, is using what's called ozone-producing UV lamp. This is the low-pressure mercury lamp that basically emits two wavelengths, 185 and 254 nanometers. And that, again, you can read the mechanism, but basically what happens is we produce the oxygen, there's an ozone produced. Ozone is produced by the 185 nanometer light, but it's destroyed by 254. So there's this kind of constant reaction of the, OH radicals, atomic oxygen present. And what happens is that generation of those active molecules basically oxidizes the surface of the thermoplastic. So every plastic is different. They may have different number of those unsaturated molecular functional groups, so they will absorb differently. Some materials are more prone to, for example, those polymer scissions, meaning I'm going to cut literally the thermoplastic polymer chains long into shorter ones. And there are consequences of this. So the UV ozone light, and you'll see that as well today, it nicely oxidizes the surface. And nice thing is that whichever the functionalities we form, they all will make the surface hydrophilic. Different setups, even the commercial ones, you'll see that there are different positions from the light source. And again, I want to caution you that it's really important to pay attention how far from the light source you're going to position your sample. Because you can see here the data we published. You have a UV source, UV ozone producing lamp. You have different levels. The lamp is positioned on top. So where you position this, you see the different degree of oxidation here as presented by the presence of the carboxy functionalities, those COOH groups. So level 1 is the closest to the light source and produces the largest number of those oxygen-containing functionalities, which makes sense. But again, when you develop your protocols, you develop your QC protocols, you need to pay attention that you always position your material the same level from the light source. This is just, I realize, very busy slide, but basically illustrates how complicated that actually photooxidation is. And there are a lot of functionalities. Those could be, carbonyl, methoxyl radicals, and you can really analyze it when you're organic chemists. You can form peroxides. There are definitely scissions for some polymers. One that we really pay attention to is those carboxy functionalities, and I'll tell you later why that is. The best way to evaluate the composition or how that composition changes is using X-ray photoelectron spectroscopy. This will give you the really intricate information about the composition of those different functionality, whether you have more aldehydes or ketones, whether the esters are the dominating functional type of groups. XPS only penetrates about 2 nanometers. So when you activate surface, when you perform photooxidation, you're really working with that top monolayer. So you need a tool, an analytical tool that will predominantly test that top layer, monolayer, not the bulk material. So this is extremely important. If you want reliable data, XPS is your method of choice. So some polymers are susceptible to scissions, or what we call, you can ablate the polymer if you expose to UV light for a long time. And this is the same process when you were a kid and you left your plastic toy outside for the summer, and at the end of the summer they lost the color, right? That's basically what it is. We bleach it by the sun. We ablate of the surface by constant exposure to the UV rays. This UV lamp, that's what it is. It's just we do it fast in a high intensity mode, so we can do this activation pretty fast. So you can see here that PMM, especially when you expose for 15 minutes, very close to the UV ozone producing light, you can see the ablation of the polymer. If I run the profile of the surface exposed through the what's called TEM grid, you can see that you can even form 400 nanometer trenches, so to speak. For microfluidics, that may be absolutely of no consequence to you, okay? But when we're working with nanofluidic devices, that could be absolutely essential to know that this process is taking place. Because if as Sanguk was talking yesterday, you're producing 20 nanometer structures, ablating this and as deep as 400 will erase basically your fabrications, right? So those are things that you need to consider. And there are some polymers that are absolutely, resistant to this, like polycarbonate. COC, a little, quite negligible. As another consequence of treating the thermoplastic with UV light is when you do have those scissions, you are lowering glass transition temperature. Shorter polymer chains have lower glass transition temperature, longer ones much higher. So if you think of basically sort of creating the scission on the top monolayer, or very top layer, again, you have to take this into consideration and maybe use this to your benefit. we actually, when we do UV modification of thermoplastics, then you can lower that temperature at which you do assembly, because the top monolayer of chopped polymers will act as a glue on your substrate and your cover plate. So this is not a deal breaker by any means. This is something you have to consider, especially when you're working with nano devices in a nano domain. Again, for microfluidics, maybe that does not make a big difference, but you have to assess that before when you evaluate your application. So again, the same type of functionalities we see on polymethyl methacrylate, cyclic olefin copolymer, COP, PC. There's a slightly different mechanism, and you'll find this in the back section of my notes. This is for this conversation. It's not relevant, but we always we look at this, so you can see this is the XPS spectrum before oxidation, before photooxidation for COC. And this is so nice to see and compare it after, because if you look at the structure of the COC, there is no internal oxygen in there, right? So you can see a clear arise in those oxygen-containing functionalities. If we look at the carboxy functionalities, about 10% of all the functional groups that we receive after photooxidation. And those, if you look at those different functional groups, and you evaluate what's called pKa value. It's this dissociation constant where in chemistry, we know at which pH that molecule will either lose the proton or gain the proton. At what pH this molecule will be negatively charged in terms of for the carboxy functionalities, for the COOH group, and at what pH it's going to be in neutral form. So that's very important metrics. And you can, for the carboxy functionalities, that pKa value is around 5, meaning that when you're working in your physiological sort of buffer at pH 7, the surface of your microfluidic, it's going to be negatively charged. If you measure, if this could be of high consequence when you do capillary microfluidic capillary electrophoresis. Here you're looking at the measure of the electrophoretic mobility, or EOF, versus versus pH of the buffer for different type of polymers, modifying pristine, as we call it, and activated by the UV. And you can see that the EOF starts picking up for all of those right at this value of 5. That's basically the representation of the pKa value. There's another reason. So among those different functional groups, only carboxy COOH group, those carboxylic acids will be only protonated or deprotonated. The pKa values of alcohol ketones are very high. We won't be able to reach those values in those microfluidics in a typical aqueous buffers. So that's why carboxy functionalities are so interesting. We can use them as charge modulators and also anchors for covalent attachment of other molecules. And now we're gonna kind of transition to that how to utilize those carboxylic groups, those COOH groups for attachment. So just as a side, sometimes it's really important to actually measure how many of those COOH groups you have. And there's a nice and simple method to using this toluidine blue all molecule that is positively charged, and forms one-to-one complexes with negatively charged COO in that case, right? So if you perform the assay, you know you can measure colorimetrically, you can measure what's the concentration of your TBO in solution, and that will be basically indicative of number of moles of the COO negative functionalities on the surface of a microfluidic, of a thermoplastic. So when you do, and I kind of touched on this, that depending on, composition of your material, the number of those oxygen-containing functionalities, or COOH, let's just narrow down the conversation to that, will be different. And what we've noticed is that there is the composition dependency on the functional groups. We've noticed here, PE is the complete extreme, right? When you consider that the COC is a composite of polyethylene and the norbornene. So here are the level of COOH after photooxidation for polyethylene. Not much really, but if you look at the COC 6017, it's much higher for this blue bar, basically, which tells you that it has 82% of the norbornene. Basically, higher percentage of norbornene, we've noticed you have higher concentration of the carboxy functionalities on the surface of the polymer. So again, this is something to pay attention. Don't just say you're gonna use COC. Be specific. What type of COC you're gonna use, because it will affect ultimately the application. COC and COP, they are being activated pretty much the same way. We see the same trends for these two materials. But I want to bring your attention to one thing. Usually, the data that I showed you in those past slides, usually we perform the UV activation with the UV ozone light for 15 minutes. If you look at this very graph here, here's a COP activated for 13 minutes and then COP activated for 26 minutes. And like you can see clearly here that, well, the longer exposure time does not produce more of those cyclic acid, carboxylic acid, right? Actually, if you may argue, you can see the downward trend. And that's exactly right. The bottom line is more exposure to UV ozone light is not always better. There is a threshold that you cannot pass, because what's happening, you start basically, cleaning and destroying the functional groups that you already created. So here you're looking at the data secured with what's called super resolution imaging, super resolution microscopy, where each one of those crosses represents where the carboxy functionality was converted into via this amide linker with the dye. So we're observing basically what is the density of the dye molecule, single dye molecule on the surface. And that is indicative of a density of the carboxy functionalities. And you can see that there is COC for up to 15 minutes. You see the number of those crosses is increasing, reaching about quite a high density at 15 minutes. But when you pass 20 minutes, it decreases. And that's exactly what happens is you cross that threshold of 13, 15 minutes, and you start destroying the functionalities. So this is very important. More is not always best. Whichever conditions you're gonna use though, what we've noticed is that those functionalities are the modification activation is really stable. Here you have results, and we measured the water contact angle for 30 days, and really there are some variation, but overall this is very stable surface, especially in contrast with the PDMS that we certainly know how to make PDMS hydrophobic and keep it hydrophobic. So this is in a vast contrast to PDMS. Thermoplastic, after UV ozone activation, those surfaces are stable for a long time. So what can you do with this? Well, you can attach oligonucleotide using this EDC NHS chemistry, what we call it. What basically is using those carboxy functionalities, creates an ester, succimidyl NHS ester, and hydroxyl succimidyl ester. And then when you bring the molecule with the lysine or primary amine, they will create a beautiful covalent attachment forming this amide bond and will not go anywhere. So you can attach oligonucleotides, you can create microarrays. This is an example of that. You can bring protein molecules. They have quite a few lysine in their structure that you can utilize. If you do it carefully, you will not affect the activity of the protein molecule. Another thing is just consider when you're working with high aspect ratio structures. Remember, I told you that there's this ablation on PMMA. Well, the problem is that you're changing the absorption coefficient of that polymer. So you're gonna see what's called kind of shielding effect. When you modify the surface on top, it will prevent basically UV light getting into the depth of your microfluidic high aspect ratio channels. You can see this is the intensity of the 150 micrometer deep channel for PMMA. This bright thing indicates the fluorescent dye. So you can see that here's the bottom of the PMMA. This is the modification, the gradient. This is the COC. COC is much better for the UV, accepting UV activation for high aspect ratio structures. So here's the example of creating microarrays using thermoplastics. Truly here you're imaging the molecules that are hybridized to your microarray. You can form those hybridization type of assays, immunoassays in enclosed structures. This is the device with the post that give you additional surface area, will basically extend the dynamic range. You can isolate or you can detect wider dynamic range of the molecules. As I mentioned, you can attach the proteins. Antibodies are proteins, right? So that's how you develop affinity isolation assay, by covalently linking antibody to the surface of a microfluidic, and you can perform, you can target specific protein in a plasma, or you can target specific cell with some specific antigen on the surface by creating antigen antibody interactions in a microfluidic format. And by changing that antibody, you can design assay for different type of cells, for rare cells or not rare cells, but you can isolate them in an elegant format. I want to just as a practical aspect, when you buy antibodies, those are very often stabilized with sodium azide. Before you do perform this EDC NHS attachment to the surface, you need to eliminate the sodium azide. Sodium azide will also react with your surface, and because it's a very small molecule, will really decrease the coverage of your antibody on the surface by competing in this EDC NH reaction. So always make sure that you remove either sodium azide or sometimes the protein antibodies are stabilized with bovine serum albumin, you have to find a way to remove it. And there's plenty of ways to do this, so this is not a big deal. So, we know how to directly attach protein to the surface. Now we can design that chemistry, and this is exactly what we did here. We created what's called cleavable linker between the antibody and the surface. You introduce the linker that you can cut. In this case, this is this uracil that can be enzymatically cleaved with this user enzyme. And we published nice work on this. It's an elegant way of releasing the molecules or releasing cells from the surface for the downstream analysis. You can use actually blue light or violet light if you design a linker containing coumarin functionality. And you can, after isolation of the biologicals on the surface with the coumarin functionality here between the antibody and antigen, then you shine the 400 nanometer light on that fluidic, you can again release the molecules, which is really, really elegant. A lot of our collaborators, they seek that kind of analysis where you isolate biomolecules or cells, or I'll talk about the later exosomes, for example. We isolate, we present them with a tube of material that's isolated, and they can perform benchtop type of analysis. So I put a lot of attention on carboxy functionalities that give you ability to create surface that's either neutral, or if you go to high pH above 5 to negative. But you can turn the table, and actually you can attach the amine molecule, and at that pKa value or pKb for the basis, you can actually turn it around and make it either neutral or positive. So there are ways of designing the charge, and here's the reaction where you basically end up with the covalently attaching the diamine molecule on the surface, and you can see how this affects charge density, type of charge, whether it's again positive, neutral, or positive. It will affect the zeta potential. You will reverse the electroosmotic flow that you typically see in microfluidics. Their surface is negatively charged. So that's a very simple, elegant way of doing this. Remember, I mentioned, normally we want the surface to be hydrophilic to accommodate all the, aqueous-based sample processing, right? But you can, again, go the opposite direction. Maybe there's an application like this generation of the reactions in droplet where you would like to have this actually hydrophobic. So there is a way of doing this, and by attaching this trichlorosilane to the surface, and we just without the shame, we borrow from the glass chemistry, which I tell you, when we started and when I started in Dr. Supper Group, we knew there's a lot of nice chemistry developed for glass, but there were generations of researchers in Dr. Supper's group who worked on developing those chemistry to accommodate, to work with thermoplastics. So yes, you can see you can create the surface that's actually in a thermoplastic that's non-wetting, and you can create the beautiful uniform size of the droplet of water in oil droplets. For you friends who are still working with PDMS because of type of applications, because you're doing, for example, cell culture on a chip, and you think you absolutely need that PDMS that has the gas permeation ability, right, for CO2 and oxygen. We can talk more about this. You really don't need to stick to the PDMS. But if you do, there are ways to bond and form those hybrid material devices with PDMS and thermoplastics. And here I listed quite a few examples how this can be done. But believe me, actually, you're not stuck with PDMS even when you're doing the cell cultures. So in summary, you really need to be aware of what you want from your surface, because this designing and this, activation modification of thermoplastics is fun, but it takes time. It takes really serious attention to details, and you need to have tools that will reliably, evaluate the nature and architecture of that surface. So there is a high value in designing. There are a variety of ways you can approach your application and building of the thermoplastic surface. And I encourage you to look at this bioconjugate techniques book by Hermanson. It gave me there are a lot of reactions there that give me idea how I can actually apply this to solid phase, to thermoplastics. So with that, I'm gonna conclude. This is the army of people who actually worked diligently over the years to develop those chemistries that I just presented to you. And I think we have time maybe for one or two questions if you have any. Thank you. I don't see any questions. None of you. Okay, go for it. Yes. Maybe you can come up with those, but I would not advise that because you don't have control. When you rely on absorption, basically, everything can absorb. And even if you design somehow, where you can release that from, then again, you're releasing everything. Your assay will suffer poor purity because you have everything. You're losing the attractiveness of immunoassay, right? Where you target very specific population of cells, very particular, peptides, proteins. So this is not necessarily the ideal. And that gives you also, you don't always have a control. Okay, how strong is this non-specific adsorption? If I'm gonna process 100 microliters, let's say, of blood, okay, maybe that's gonna be still there. But if I need to up a volume to 1 milliliter, are you gonna start washing it off? So relying on non-specific adsorption, for example, the whole ELISA concept, right? Quite well, but it's a stationary, right? It's just the plates are covered with basically adsorb the antibody or antigen. You introduce your sample, it's stationary, right? It's also not ideal, but we prefer something that gives you confidence that even if I process, 10 mils of sample, I still have that covalent attachment. Of course, so absolutely. So, you can design assays that you perform negative selection first, And then you, with material that you have, then you go for positive, right? But there are ways maybe of maybe tagging those molecules for the negative selection and executing way of, transducing that mode of negative selection on a chip. Yep. Alrighty. I'm gonna welcome the next speaker.